Good afternoon, guys. Hello, four steps up. New student, we welcome you. Yes, ma'am. Okay, good afternoon. Kadir, Samyukta, four steps up. Nihariga, Amrita, Prabhu, KP, Adams, Srividya, Nandini, ma'am, Krika. Okay, hope you all doing good. Six members are there. Let's wait for 2 minutes. Still Saran yet to join. Saran, Harini, Prakalya, Rangavarma, Ashwin, Meerahul, Adams. Rajavarma. Oh, Adams, you're here. Yeah. Okay. Sorry. Okay. Yeah. Sivaditya, 10 members came. Out of 8 members. Still 8 members should come. Rajavarma, Napoleon, you're here. Yeah, hi Rajavarma. Okay. Good afternoon. AG Prakalya. We'll see for one or 2 minutes and we'll start the session. Today's session will be a little short because this is new topic. I don't want to take much lengthy time here. And within this 3:30 to 4:30 itself, you have to finish the homework, okay, and post it. And I don't encourage you to post homeworks late in the nights or after the given schedule time. Okay, so within the time frame, you please. OST is yeah. If the questions are, you can reduce the number of questions also. For example, you can write at least 2-3 questions. That's enough related to this concept. Okay. Okay then. Can't add any charts right now. Okay, meanwhile, you just answer what is plant breeding. Plant breeding introduction I already gave, right? What is plant breeding, etc. So I'll be posting one more question. What is a plant breeding? Okay, you just let me know. Actually, I wanted to include cards here. The after the live session ends, right? There will be cards will be displayed. But right now I will be posting the question here itself. Okay, try to answer it. So what is plant breeding? I gave two options here. So just see in the comment section. Try to answer. 14 members are here. Harini came. Prakalya, Harini, Ashwin. Okay, meanwhile you answered this question. Prabhu KB Harini, yeah, Harini is here. Ashwin Mehra, okay. Prakalya is here. Okay, then we can start now. It's already 5 minutes. Okay, now I already posted the question right now. That is, what is plant breeding? So plant breeding is a purposeful manipulation of plant species to get what? Is it a better yielding variety and disease resistant crops or disease crops? That's the question. I mean, it's a very simple question. I just wanted to make time goes. Okay, everyone. Yeah. Answer A is correct. Option A is correct. Correct. Okay, now let's start. So if you see here, for achieving the plant breeding, we need to have a proper strategy. Okay, I told right. This cropping system is started way back, when the human is evolved, our species, right? So the modern human evolved. This agricultural setup is evolved just around 10,000 years ago. So from then the conventional plant breeding techniques to now the biotechnological advancements, there is an array of practices we do in the plant breeding. And if we put all together, we have a step-by-step procedure. Okay. Today we're gonna see that step-by-step procedure. Last week I already gave a little introduction about it, right? Right. So I will be pasting that step-by-step procedure also. Meanwhile, you can look into your books as well. The step-by-step procedure of the plant breeding technique that we have already, I introduced last week, and I will be pasting here itself. Okay, what we're gonna discuss is step-by-step plant breeding procedures. Plant breeding procedure. Right. First step, what we're gonna study is collection of variability. Okay, so I just wanted to bring plants and show some kind of a thing. It's not really necessary, but our entire point here is to understand how can we really create a variety. Last class we discussed about a table of contents which we have memorized. We charted out the memory technique, mnemonic, to remember those varieties. It means the normal crop and its variety, disease resistant variety, right? So to achieve that, what are the steps we need to follow? Okay, those steps I will be putting there in the comment section, right? So there are 5 different steps out there. From first step to last step, we will have a very a kind of simple way of understanding. Okay. So what is it okay? Am I clear what we're gonna learn today? What we're gonna learn, is that clear, guys? What we're gonna learn is step-by-step procedures, steps involved in plant breeding mechanism, right? Yes. So am I clear, guys? You should know. Yeah, good. Okay. Is my voice audible, right? Voice and the image. Is it clear, right? Okay. So first step. Good afternoon. Tangavarma here. Okay, Tangavarma. Okay, fine. So to create any variety, right? So we need to follow certain procedure, right? So a plant, if you see this plant breeding program, right? It is carried it is carried out in a systematic way, right? It's like worldwide approved by governments and other what do you say, farmers and other whoever follows this plant breeding techniques. It is approved, and other agricultural institutes and commercial companies also, you know been approved this kind of a strategy. So it has to go in a systematic way worldwide, wherever it is. This plant breeding technique has these kind of a steps. Okay. So in this plant breeding program, what are the steps? So first step is collection of variability. So what does it mean actually? Variability here, what does it mean? Can anyone guess what you mean by variability? What we are talking about here? What is variability here? We are talking about variability in what, for that matter. We are looking into variability, right? That is good. But what exactly we wanted to get that? Variability in what? In these plants? What we are looking for? What does it mean here? So actually, like in our class, we always go by a general English meaning, and then we'll go for scientific meaning, right? So what does it variability means? What is variability in this context, at least? You can contextualize and understand, like get what in my variability here. Anyone? Variability, variable, variation characteristics of plant. Variation in genes. Good, very good. Traits, right? Characteristics or traits. Yeah. Anyone else? Difference in size. Okay. So you are talking about characteristics. Okay. Okay. So variability here, what we are talking about is, it's at the level of genes, genetic variability, right? So the variability of a plant, right? We can collect variability of any plant from their seeds, isn't it? If you take any plants, yeah, different species, varieties, variation characteristics. Yeah, correct, Amrita, and Srividya, correct, and even uh, Prakalya, correct. Yeah. So if you see here, we are collecting the variations first variabilities we are collecting. It means different traits and different characteristics that we are looking, or that we are desired, that we are interested in, right? That we wanted to see in offsprings, right? Those variabilities are present in the seeds of a plant, parent plants. So first we go around, look around, and we'll see that, yeah, this is drought resistant variety, right? This is disease resistant variety. This is pest resistant variety, right? It is high yielding variety of the same species. So we collect those seeds. So to create any kind of offsprings, first we need parents, isn't it? Yes or no? So to get the offspring of desired characteristics, first we wanted to collect the variability. Variability in the form of nothing but seeds. Variability in the form of seeds, and the entire, for example, either seeds or sometimes plants itself. Directly we collect the plants or the seeds. So this collection of variability and storage, we say, this collection we call it as germ plasm. So first we collect germ plasm. For example, we collect any seeds, right? So we sow, we store these seeds. Okay, these seeds will be stored, right? So this, either this collection of seeds or plants as a whole, to breed them for our desired characteristics in offspring, right? The first, very foremost step is to collection of variability or collection of germplasm. So germplasm includes both what plants or seeds itself. Is that clear? So here there is an important definition here, that is 1 mark, maybe 2 marks are also. What is germplasm collection? So germplasm collection is nothing but the collection, the entire collection of plants or seeds having all the diverse alleles. Alleles we already know, the alternate form of the genes, right? For all genes in a given crop. So that is called germplasm. So germplasm collection is one of the important definition you will be seeing in the plant breeding program, and that is important for 2 marks, right? Germplasm collection, right? So this is first step. So first step is to collect the germplasm or variability. Variability at the gene level. So how can we really collect the variability at the gene level? Either plant as a whole or seeds of the plants. Is the first step clear, everyone? You guys are there? Please do respond. Is first step clear? Remaining. Okay, great. Okay, great. Yeah, that's good. That's good. So let's now, so we got the plants and the seeds, right? So what might be the second step, right? So the second step is evaluation of parents. So now we are going to talk about evaluation of parents. So I told, right, second step is evaluation and selection of parents. Once we get this seeds and plants, right, it doesn't mean that the plants, every plant that we collect and every seed that we collect will show the, once we sow and when we see the characteristics, maybe 100 out of 100, maybe 50 may show the desired characteristics. So we need to artificially select those parents which have desired characteristics. So that is the second step of this procedure or the systematic procedure of plant breeding, right? So what else we can see in this second step, that is evaluation and selection of parents. So far what happened was, like, so far we collected the germplasm, right? So after collection of germplasm, this germplasm is being evaluated to identify plants with a desirable combination of characteristics. Maybe someone said height, right? So maybe a high yielding variety, right? Or maybe disease resistant variety, right? So once we see this desired combination of characteristics, the selected plants are multiplied and used in process of hybridization. It means now we got the germplasm in the first step, or the now the parents have been selected by growing them, and we will select whether they have the desired characteristics or not, right? And then we go for hybridization. Hybridization is it's a simple means of crossing. You have seen in Mendel cross, right? Mendel, how he crossed, cross pollination and self pollination. What we see is in the same way we will go for hybridization, right? So when we go for this hybridization, what we can create is a pure lines. So what we can create is pure lines of it. So pure lines, the second step of this plant breeding technique is generating the pure lines. Pure lines are created where desirable and possible. It means the pure lines which are having the desired characteristics, right? Those pure lines will be produced during this step. Is that clear? So second step is something which we already collected the germplasm, and that's that germplasm which has either seeds or plants, right, will be selected and will be used for the next step, that is hybridization. So this is how we will evaluate the parents, right? So you have to remember one thing, right? That is, this entire systematic procedure starts with the seeds and ends with the seeds. A seeds which are something which has a desired characteristics, right? Maybe A, B, C, D, right? So AB, for example, AB seeds has got desirable characteristics. So we hybridize them, and then we see these AB characteristics in offspring at the end of this procedure. So this entire procedure, even when you're writing the exam, maybe sometimes we may forget where to start, where to end. So this plant breeding technique starts with the collection of seeds and ends with the distribution of seeds. As simple as that. So first step is clear, right? The first step speaks about collection of germplasm and variability. Second step is selection and evaluation of those parents, right, for the further hybridization. Is the second step is clear? Second step is clear, guys? The second step is clear? Okay. No doubt, right? Yeah. Let's go for the third step, right? So let me paste this third step of this plant breeding technique. That is cross hybridization among selected parents. See, this plant breeding technique may be asked as 3 marks steps. The only the headings in 2 marks, or the procedure in 5 marks. So it is, one leads to other. That's why we say step by step. So first we collected germplasm. Second we evaluated the parents, right, for the cross hybridization. Third step is hybridization, right? So the third step we already studied in principles of inheritance and variation. Mendel cross. You have crossed n number of different types of plant, different types of characteristics, traits, complete dominance, incomplete, etc., etc. So here what we are using. So in the third step, what we are doing is cross hybridization among the selected parents. Now though we already selected the parents, right? So these parents has to be crossed. That's how we create the offsprings, right? What is the point? So we have to create the offspring with the desired characteristics. So if you forget this step, there is no meaning for your answer. So think once you actually, when you write the answer, you should not miss this step. So third step is cross hybridization among the selected plants. So how do we do that? What exactly this cross hybridization involves, right? So in this cross hybridization, you would have seen in principles of inheritance and variation, right? And at the same time, second chapter that we have talked about, you know, how to actually do the self pollination, how to avoid cross pollination, right? How to select, simple, how to, if you see this Mendel, what he have done, right? So he have carefully is to dust the pollen onto the desired plants. So like that, what we can do in this cross hybridization step is nothing but the desired characteristics have very often to be combined from two different plants. Nothing but two different plants we are actually combining. A has some disease resistant variety. B has got some, for example, high yielding variety and disease resistant variety. So these two parents has to be cross hybridized and to produce the hybrids. So we do the cross pollination, and then we dust. What we do is we collect. Naturally it will take a lot of time, it may kill a lot of time. So we collect the pollen, and then we dust it on the stigmas of the desired female flowers, and then finally what we get is a hybrid seeds, right? So this is how we can go for cross hybridization. Okay, is it clear? So sometimes, what happens is, the hybrids that we create, no, not always they show the hybrid characteristics. That is, desirable characteristics may not show every time. Maybe out of 100, maybe only 10 plants may show. At that point of time, what we do? That is the second. That is the next step. Yeah. So quickly again recap, right? First we collected the seeds, germ plasm, and then we selected the and evaluated the parents. And third step we hybridized parents. It means cross pollinated by taking the pollen from the male anther pollen and dust it on the stigma, female flowers, and we create hybrids and we sow them. And the technical point here, what we have to keep in mind is sometimes or most of the times the desirable characteristics plants may not be, the plants may not show always the desirable characteristics. So out of 100, only 10 may show. So to get those pure lines, so the desired plants, what we have to do is the fourth step. That is, we have to select those hybrids, right? Out of 100, only 10 got hybrid or desired characteristics, right? So those has to be selected. So far hybridization is clear, right? So when we see this fourth step, that is selection and testing of superior recombinants. So what happened was, like in the previous step, maybe out of 100 or out of 1,000, right? Only 100 plants may show the desired characteristics. So we will be selecting only those 100 plants, not the other 900 plants. Right? Yeah, exactly. So again, see, the third step led to the fourth step, right? That's that much easy. But once you see as an without understanding, if you see this page now, you will get headache. Oh, what is this plan reading? Why we have to read? Is it necessary? Can we skip this? Like, there are many ideas you may get. But once you read in this perspective, it will be very easy. It's a piece of cake, right? So the first step is something which is we are concentrating on those 100 plants which became hybrids, the superior, the recombinants, the hybrids, superior recombinants, right? So those superior recombinants we will select and we test, right? We will be selecting those plants and will be tested for their superior recombinants. And how do we test for the superior recombinants? Again, this selection process is a crucial to the success of breeding, isn't it? Because once we get the hybrids, these hybrids only, the hybrid seeds only the main aspect of this entire plant breeding technique, right? Steps. So this is very important and crucial step. So this step yields plants that are superior to both of the parents. It means one might be drought resistance and one might be high yielding variety. So both the traits, both the characters should be expressed in this hybrid. So those plants will be selected. And once we select these plants 100 out of 1,000, these are self-pollinated for several generations, right? So several generations, what we do is we self-pollinate. So self-pollination leads to one of the condition called homozygosity. What do you mean by homozygosity? What do you understand by term homozygosity? Yeah, identical genetic makeup. Is it clear? Anyone? Homozygosity. Just split the term. Zygosity. Yeah, similar. Similar in what? Genetic makeup. Pure lines, for that matter. Are you getting? State of uniform. Order school uniform, right, Adams? Yeah, uniform genetic matter, or you can say similar genetic matter. Yeah, Adams, good. Sridhya, good. Obviously, good. Same kind. What same kind? Prakalya. Exactly. Kritika, good. Same kind, yeah. Prakalya, good. Okay. Remaining, are you guys out there? Are you guys sleeping like this? Remaining, others, Ra? No, ma'am, they are listening. Hey, guys. Jean, yeah. Others. Okay. So now you understand the fourth step, which is something which we create homozygosity. So once we collect these hybrids, we select these hybrids, right? Yeah, Kadir, right? Yeah. Same kind of a gene. Yeah. So once we yeah, Raj Verma, correct. Shivaditya, very good. Okay, no, ma'am, no one is sleeping. Homo means same. What is this? Okay. Yeah. So everyone replied. Very good. So once we get this hybrids, right? 100 out of 1,000. Yeah. Someone wrote the possession of two identical alleles of a particular gene or genes by an individual. Excellent. Correct. Okay. 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 Good. Good, Pritiga. Yeah. Fine. Dharani Kumar is using actually. Okay. Let's move on. So in the fourth step, what we are doing is selection and testing of superior recombinants. Once we get the hybrids out of 1,900, uh, out of 1,000, hundreds, 100 plants show these desired characteristics. So these desired characteristics will be again self-pollinated for several generations. So when self-pollination occurs, it creates a homozygosity. And when we are talking about animal breeding, we'll talk about inbreeding depression that will be touched upon. It means creating homozygosity, how it will be positive as well as negative. But when it comes to this homozygosity, right? In fourth step we are trying to create the homozygosity so that the characteristics will not segregate in progeny. We see the law of segregation, right? In the law of segregation, what we studied is the parental genes, right? Any one of the recessive trait may express in the progeny, right? So that will not be present when we go for this self pollination, and that will be achieved in the fourth step. So is that fourth step is clear? So what we are creating is a superior recombinants. We are creating and isolating. We are trying to capture them. So that is the whole goal of this entire plant breeding. Shall we go for the last step? And I will. We can break for the day, and you can start doing the questions related to this topic. Okay. Very good. Okay. So the last step is testing, release, and commercialization of new cultivars. Right. So if you memorize, right, if you remember, right, this entered these all types, right? So what we are going to do is finally the last step is we are creating the cultivar, right? So cultivar with the desirable traits or characteristics that the superior recombinants that we got in previous step. So this would be the last step of this plant breeding, right? So what does it contains? So it contains, so you can see that. So the first step started with something with a collection of seeds. Now the last step is going to end with the seeds, which is nothing but distribution of seeds. So once we did this selection and testing of superior recombinants, we go for its testing. So we got the homozygosity, right? Homozygosity we created in that particular hybrids. So again, these lines, right? These pure lines that we created are evaluated for their yield and their agronomic traits of quality, disease resistance, etc. So what we're gonna do is we will be again showing them for the many generations, and we will see whether they are holding the traits or not. So we'll be testing these superior recombinants, right, for their yield and agronomic traits of quality. The quality which or they possess in by in the seeds, what they possess is nothing but disease resistance or other quality that we gonna see. So that is what one of the step that includes within this step, that is testing. And once this testing is done, now nothing but the evaluation is done, right? How we do is nothing but again we will be growing in this research fields, and we will be recording its performance also, how they are performing in different seasons and different conditions that we actually looking in that particular recombinant or hybrid, right? So that's how we test it. And once the evaluation is done, we will be testing these seeds in the farmers fields. So it has to go to the farmers fields, and we will be testing these seeds that are we cultivated. This cultivars will be tested in the farmers seeds fields, and will be growing for several generations and several rotations. So we wanted to make sure that maybe if we create this cultivar for South Indian soils, right? South Indian different agricultural soil, we wanted to sow these seeds and test these hybrids in different locations, not only in Gobi. We have to see in Kakinada also, we have to see in Hyderabad also, we have to see in Chennai also. Then we will get the complete representation of this performance of these hybrids. Once they perform well, what we can do is we will be releasing for the local farmers, right? And then we can commercialize it. Commercialization is nothing but we can sell it. So in that way, what we are doing is we are actually distributing these hybrids. So this is the final step of the plant breeding technique. So this is one of if you can I think you understood the fifth step, right? Is the fifth step is clear? The fifth step is clear, guys? Yeah. Okay. So what happened was like we started with seeds, right, and ends with seeds. Start starts starts with seeds, ends with seeds. So quickly recollect what are the steps we have learned so far, right? So what are the steps we learned so far is nothing but first one, collection of variability. So in this variability collection, we talked about germplasm, and next one is evaluation and selection of parents, isn't it? So we will be evaluating and we will be selecting the parents for the further hybridization. So the third step is cross hybridization among the selected parents. Maybe A has disease resistance, B has high yielding. We will cross them and create recombinants, right? And then fourth one is selection and testing of superior recombinants. Once we collect these parents, we what we do is we go for homozygosity. It means we create superior recombinant. This superior recombinants will be created by simple self pollination for many generations, and we create pure lines to avoid segregation, right? And then finally the fifth step is testing release and commercialization of new cultivars. So once we done with this, so we have to test it in the laboratory, scientific fields, and then we will go for the farmer fields for many generation different locations, and then finally we release this cultivars in the market. So this is how the plant breeding technique works. I hope it's clear, right? So let's utilize this last 20 minutes for your homework. And regarding homework, I'll give questions now itself. What you can do is, I want to waste time, your valuable time, and you should not sit with mobile the whole day. So homework, you can relate it to this plant breeding technique. Seventh question: briefly describe various steps involved in plant breeding. Okay, so seventh question. You just write the seventh question itself. Question number 7. That alone enough for today. That's it. No more discussion. Is that clear? Okay, guys. Is that clear then? We will meet in the next session. Okay, this session is we are ending just 15 minutes early, right? Just 15 minutes early we are ending because I wanted to make sure that you will do this homework as well, right? So thank you guys for your response. It's really, I'm really amazed to see your response. Very happy. Yeah, and thanks 4 steps up for joining here. Thank you, Niharika. And yeah, thank you guys. Okay, ma'am. Thank you, Nandini, ma'am. We are ending the session, and you please complete the homework. Yeah, yeah, me too. Waiting for the next session, and we'll come for come. I will come up with something more interesting. Yeah, thank you. Bye, guys. Jai Hind. Bye, bye, bye. Saran, bye.